First of all, I'd like to thank Alan for organizing this wonderful symposium and for our guests for coming all this way to give wonderful talks. Thanks. So I'm going to talk today about a novel system for abnormal chromosome correction that we uncovered in human cells, and that is ring chromosome correction during induced pluripotent stem cell reprogramming. And as you can imagine, this wasn't something that we set out to investigate, although I have studied genome instability in the past. But we stumbled upon it when we were studying this disorder, this human disorder. So now we're going to get away from yeast a little bit. I think Stan's probably going to talk a little about human as well next. We're studying this disorder. We're studying for many years lissencephaly, which is refers to the smooth brain. As you can imagine, although the brains are very similar in organization, brains miss the normal gyri and sulci of primate development. And so the children that have this have severe mental retardation, seizures, and early death. There are many forms of lissencephaly, but one of the most severe is Miller-Dieker Syndrome, and that results in a completely agyric brain, here shown in this MRI, and some recognizable craniofacial dysmorphisms that a medical geneticist, of which I'm one, could easily recognize. All children with Miller-Dieker syndrome have about at least a 1 megabase deletion on the tip of chromosome 17p that includes more than 20 genes, including a gene called LIS1. And although I'm not going to talk much about evolution, since evolution is the title of the seminar as well, I just want to point out that we knew nothing about what LIS1 did until its function was uncovered in the bread mold Aspergillus nigellans, and was found that it interacts with dynein and important for dynein regulation and function. So that's my little foray into evolution for my talk. So what we wanted to do, although we found a lot out about the phenotypes and the mechanism of action of LIS1 using mouse models, we were interested in trying to make human cellular models of this disorder. And so in this case, we were taking advantage of the recently described and now ubiquitous method of induced pluripotent stem cell generation by cellular reprogramming. And as you all know, this was developed by Shinya Yamanaka, who won the Nobel Prize for this several years ago. And the concept is you take a skin cell or any other cell from a patient, and by transfecting transiently cells with 4 to 6 transcription and other factors, those skin fibroblasts will be reprogrammed into pluripotent stem cells similar to ES cells. And these cells can then be studied, differentiated in vitro to any sort of cell type that one wants to study. Let's say, again, the disease is lissencephaly, then one would want to make brain cells, study those cells, and see what can go wrong for a cellular disease model. In addition, if the genetic defect is corrected, it could also be used for cell therapy in the patient if there is a cell therapy modality for the disorder. So Marina Burstein, a postdoc in my lab when I was at UCSF, decided that she wanted to make these models. And so what Marina did was to take 3 different patient cell lines with Miller-Dieker syndrome. And being interested in the history of Miller-Dieker syndrome, we wanted to reprogram this patient's line. This patient was born in the 1980s, and a karyotype analysis indicated that the child had ring chromosome 17. And this is the first indication that anything could be wrong on 17 for Miller-Dieker syndrome. And so a number of other investigators then looked to see in other patients with Miller-Dieker syndrome whether there was something wrong on chromosome 17. And in fact, all patients had a deletion of the P arm of chromosome 17. So this patient led then to the identification of the genetic cause of Miller-Dieker syndrome, these heterozygous deletions of 17p. So Marina reprogrammed fibroblasts with a ring chromosome patient and 2 typically deleted patients. First of all, let me tell you a little about ring chromosomes. They form when the telomeres that normally protect the ends of the chromosome are lost or inactivated, allowing a chromosome to form a ring. And this could then result in both genetic and structural problems. Structural problems because ring chromosomes are difficult to undergo division, and genetic problems if there's any loss at either end, as there is in Miller-Dieker syndrome, resulting in haploinsufficiency in those problems. So the instance is quite rare, but it can occur in every chromosome, and there are human beings that have ring chromosomes in all of the autosomes as well as the X chromosome that have been described. So these are known. Now, one of the things that happens is that there's unstable behavior of ring chromosomes during mitosis. There's a failure to pair with homologous chromosomes, frequent ring loss leading to monosomy. There could be the formation of dicentric rings, secondary ring derivatives, and the formation of anaphase bridges leading to genomic instability. And because of that, there's an increased rate of aneuploidy and high cellular death rate in cells in culture in the patients themselves. In fact, patients that have no deletion resulting in any other abnormal phenotype almost always have small size. So the growth rate is reduced in patients that have ring chromosomes, and that's almost universal. So we were able to collaborate in San Francisco with the Yamanaka lab, and Marina was able to reprogram the patient fibroblasts from each of the 3 lines, one with the ring and 2 typically deleted lines, into iPS cells. And those were proven to be iPS cells because the cells were all able to make the endogenous factors that indicate that they're iPS cells listed here. Also, by in vitro and in vivo methods, we're able to show that all the cells could contribute to all 3 germ layers, mesoderm, endoderm, and ectoderm. So in fact, these cells were pluripotent. The ring chromosome line, as well as the 2 typically deleted lines, all reprogrammed at about the same frequency. And so when Marina first examined the fibroblast, of course, the ring chromosome, as well as the typically deleted lines, all had a reduction in both LIS1 mRNA, the gene that's at the edge of the deletion, as well as another gene coding for 1433 epsilon, YWHAE, in 50% levels. And the fibroblasts also had 50% levels of DNA. However, after reprogramming, although the typically deleted lines had 50% reduction in LIS1 genomic DNA, the ring chromosome lines now had a normal level of LIS1 genomic DNA. They had the normal level of LIS1 mRNA, and they also had rescue of to normal levels of production of LIS1 and 1433 epsilon. So in the fibroblasts, here's the level of wild type LIS1 and 1433 epsilon. The typically deleted lines and the ring chromosome line had 50% reduction in those proteins. However, after IPS formation, the typically deleted lines had 50% reduction in those proteins, but the ring chromosome line now had, again, correction to normal levels of these proteins. And so the reason that that occurred, this is published, so I'm just going to summarize some of the data really quickly, is that the ring chromosome that was in the fibroblasts, after reprogramming in several different clones, the ring chromosome was lost and replaced by a completely normal chromosome 17, so that there seemed to be 2 identical copies there. And this occurred sometime around passage 9. So this would just show the frequency of the ring chromosome karyotype in doing karyotypic analysis in the fibroblast at 2 different passages. And you can see that almost all the cells have the ring chromosome phenotype. After reprogramming, the majority of the cells, the vast majority of the cells now had a normal 46XY karyotype. So how did this correction occur? Or can we look at it by other molecular methods? So what Marina did was use SNP genotyping to look at copy number variants. The ring chromosome fibroblast, this would be all of chromosome 17, the P arm, the centrosome, the Q arm. And this cluster of salmon-colored dots shows that there's a 50% deletion at the tip of chromosome 17P in the ring 17 fibroblasts that was now corrected after in the iPS cells that were made from there. And iPS cells were made from the 2 typically deleted lines, and they maintain their deletion, again showed by the salmon-colored region at the tip of 17P. It's an important experiment because one possibility is that there are genes in this region that, when deleted, would not allow reprogramming to occur, but in fact, since they were able to be reprogrammed, that suggests that that's not the case. In fact, the deletion in this MDS3 line is very similar in extent to the ring chromosome line. So it doesn't seem that there's a genetic cause for this as well. So Marina then tried to think about what potential mechanisms could have occurred. And 2 occurred to us that were easy to test. One is in the original cell with the ring chromosome, there would be loss of the ring. Remember, I told you there's aneuploidy in the ring chromosome lines and in patients. And then by somatic non-disjunction, there would be complete duplication of the normal chromosome. Another possibility is that there would be a double-strand break occurring in the ring chromosome, and after mitotic homologous recombination, there would be then repair of the tip of the chromosome using the other normal chromosome as a template. Well, this is relatively easy to test, especially with the SNP arrays that we had before, because in this case, the entire chromosome would be homozygote because it would be duplicated. And in this case, only the tip of the chromosome would be now homozygote. The rest of the chromosome would be heterozygote. We could use the SNP genotypes that I showed you in the previous slide to test that out. So here just shows the results from chromosome 17. And this would be from the fibroblast in the ring chromosome patient. The representation here is for the 3 potential genotypes. So SNPs are either A or B. You could be heterozygous AB, homozygous AA, or homozygous BB. And in the ring fibroblast line, there was a mixture of all 3 genotypes. In the IPS lines derived from the ring chromosome patient now, they're completely homozygote, either AA or BB. While in the 2 different typically deleted lines that were made into IPS cells, those IPS cells had a similar distribution of heterozygous and homozygous genotypes. Those patterns were also seen in every other chromosome in the IPS cells from the ring chromosome line, the fibroblasts, or from the typically deleted patients. So it seems then like the mechanism of rescue is that in the fibroblasts where there's a ring chromosome, during reprogramming there's loss of the abnormal ring chromosome and duplication of the normal chromosome to restore a normal genotype. The same mechanism works for other chromosomes. So in this case, we looked at ring 13, 2 different patient lines. And again, the fibroblasts had ring 13, and multiple IPS clones derived from those fibroblasts from 2 different patients with 2 different rings now had normal karyotype and normal distribution of genes. And by using the SNP genotype that I showed you before, also rescued by uniparental isodisomy. So in summary then, for this part of the talk, there was ring chromosome correction in IPS cells that involves non-disjunction, ring chromosome loss, compensatory uniparental isodisomy, and selection. So what we think happens is that occasionally a cell loses the ring chromosome as it's going from fibroblast to the very proliferative state that's occurring during reprogramming. And in early passage then, if some of those cells lose the ring, they may die. But at later stages, when the ring chromosome is lost and then reduplicating the other chromosome, those cells will then be selected. And after time, either the ring chromosomes are lost or the normal karyotype is now overgrown in the cells, and it's again due to uniparental isodisomy. So you can correct then the ring chromosome defect in fibroblasts during reprogramming into induced pluripotent stem cells. So again, the Miller-Dieker syndrome can be modeled. Ring chromosomes are frequently lost in IPS cells, although I only showed you information for ring 13 and ring 17. Others have tried to model ring 14 and ring 21, and they've also found similar phenotypes in that the uniparental isodisomy rescues those ring chromosomes. The duplication of the wild-type chromosome rescues monosomy and compensates for the ring loss. Compensatory uniparental isodisomy restores a normal karyotype and the normal copy number of deleted genes. We think dynamic mosaicism in the IPS cells leads to the preferential survival of the karyotypically normal cells. So that's all well and good for the 1 in 50,000 patients that have ring chromosomes, but we're interested, I'm a human geneticist, in seeing if this may have broader applicability. It turns out that you may be aware that chromosomal abnormalities are actually quite frequent. In about 60% of spontaneous miscarriages, 4 to 11% of stillbirths, 5 to 7% of neonatal deaths, 0.5% of live births, and of course, low frequency of ring chromosomes are found. So it's actually quite a significant abnormality throughout the lifespan. Now, if you think about correcting varieties of mutations in cells for the purposes of regenerative medicine strategies, then one way to do this, if you have just point mutations, is that you could take the somatic cells with the disease genome, that is small deletions or point mutations, reprogram those cells with the disease genome into IPS cells, and then correct the disease genome with a variety of different genome editing tools such as CRISPR or TALENs, and now one will have mutation-corrected IPS cells to give back to the patients. But in the case of chromosome aberrations such as Miller-Dieker syndrome, how might you do that? And so what we're thinking one could do is perhaps if a cell has large deletion or, of course, ring chromosomes that we've shown, then perhaps during reprogramming and selection and screening, you can correct the IPS cells if you make the large deletions into ring chromosomes. And then the ring chromosomes will either be lost during reprogramming or during the propagation of those cells. And of course, there's a lot of problems before we could ever consider using this, and I'm not even attempting to say when we do this. This is just a thought experiment at the moment, because there are a lot of issues that might result in problems such as genome instability, imprinting, or recessive mutations, but I think some of those can be corrected at least by the genome editing. And so one example, I don't think Taehan's here, but Taehan Kim is a postdoc in my lab that's trying to do this. He's trying to use genome editing to insert LOXP cassettes into the ends of a chromosome that's deleted, in this case the Miller-Dieker Syndrome chromosome region. The cassettes are containing a fluorescence marker, as well as the cDNA for another fluorescence marker, TD tomato. On the other chromosome, there's a fluorescence marker and the promoter and ATG for the TD tomato construct. These are LOXP sites that are also introduced in the same orientation on each end of the chromosome in two genes that appear to be not responsible for haploinsufficiency. So once those are introduced, then Taehan's introducing Cre recombinase with the thought that if Cre recombinase will properly recombine and form a ring chromosome, these cells will then express TD tomato, and these can be selected even if it's a rare event. So Taehan has gone through all this experiment. He actually has some TD tomato positive cells now that look like they're promising candidates for having a ring chromosome. The problem is that they're pretty low in frequency, so we're trying to find ways to purify those cells so we can do karyotype and make sure they do in fact carry the ring chromosome. And then what's going to happen is if he does have cells that are TD tomato positive generated from the LOXP insertions, a cyan and venous positive cell that now becomes a TD tomato positive ring chromosome containing cell, then further propagation will result in the loss of that ring chromosome, loss of TD tomato, and then perhaps duplication of the normal chromosome to rescue that phenotype. Now, this may happen during in propagation of the iPS cells. We're hoping that that's what would occur. If not, we're going to have to do this correction to form the rings in fibroblasts or other somatic cells. And in addition to trying to correct chromosome deletions, we're also thinking to try to use this to actually model ring chromosome defects. So there's some phenotypes that you might like to examine in a cellular model. And one of those is ring chromosome 14 syndrome, which even if there's no genes that are deleted in ring chromosome 14, there seems to be a problem with gene expression on that ring, and the children have very severe phenotypes, particularly epilepsy. So one would want to make a model of this, but of course this correction mechanism for ring chromosomes has so far not allowed this to occur. So the idea would be if we can introduce LOXP sites into chromosome 14 at the ends, can we make an inducible model of ring 14? And then finally, can we correct extra chromosome copies, as Alan was talking about, such as trisomies, by perhaps, again, taking one of the extra copies, say in trisomy 21, introducing LOXP sites in the end, forming a ring, and perhaps growth selection will allow us to lose that ring and restore a normal diploid complement to those cells in culture. Then the idea would be that these cells can be used either as models or perhaps for regenerative medicine strategies in the future. So again, what I've tried to tell you about is there's a novel mechanism during reprogramming for the rescue of ring chromosomes. We're going to try to exploit that for making disease models for rings, as well as for potential cell therapy in the future. And I'll just acknowledge the people who did the work. I pointed out Marina and Taehan's major work. Again, this is a collaboration with Shinya's lab, particularly Yohei Hayashi in Shinya's lab. Other collaborators at UCSF also helped in this project as well. So thanks very much. I'll be happy to answer any questions. It's on my lap. Yes, Phil.
So it sounded like what you were saying is that the ring itself destabilizes in cis at chromosome, right?
Yes.
And
Keep on.
yeah, and were you suggesting that the reprogramming regime itself destabilizes the ring over just mitotically propagating the cells in culture?
It's hard to say. So did everybody hear the question? Is it the reprogramming itself that's causing the instability, or simply the propagation of iPS cells? So we won't know until we try to do the experiment. So the experiment that we're doing now, of course, is to do this ring chromosome formation in iPS cells. So we think that it's the rapid proliferation of iPS cells with a very, very short cell cycle very, very short G1, they synthesize DNA and actually then just divide. So that rapid cell cycle is impeded if one has a ring chromosome, and so the loss of that will then allow those cells to outcompete the other cells. So we think that's one potential mechanism of how that could occur. Another possibility, though, is we make the ring chromosomes, and the ring chromosomes are stable over many, many generations, in which case that would suggest it's due to reprogramming. So if that's the case, we now can make other somatic tissues out of the iPS cells and then reprogram them and see if that's how that's happening. But the key thing is to have a marker, so cells that have the ring chromosome with TD tomato that we can fish out of the whole population, and ones that are losing that so we can see how that's happening. So the question is a good one. Hopefully we'll find out soon. Yes,
Give you a sense of how long 2n minus 1 cell can it divide, or is it actually unable to divide unless it shows some non-disjunction?
So how long will it take a 2n minus 1 cell to divide? A lot of it would depend on the chromosome. Presumably it will take a relatively short period of time where it can be 45, have a complement of 45. The exception would be the X chromosome, because 45X, those cells can actually propagate quite well, although not completely well. So it may be that it's happening all at once, that you lose the ring chromosome and duplicate the other chromosome. It could be that the duplication is occurring first and losing the ring chromosome. Until we have a way to look at those cells by sorting or other mechanisms that have a TD tomato positive cell, we won't be able to address those. So another possibility, of course, for doing these experiments is we can find more about the mechanism of how this is occurring, and maybe even pathways that might be responsible for the loss of the ring chromosome. Yes?
So if you have lines with the ring chromosome and you partially destabilize the spindle with drugs, do you lose the ring chromosome?
Can you destabilize with drugs the spindle in a way to change the kinetics of what's going on? This is, yeah. So I think in general, in iPS cells, if you slow down the cell cycle by a variety of means, it's going to impede the pluripotent state. So that's.
What about just the fibroblasts?
With the original, with the ring chromosome?
So they probably grow slower. We didn't measure how fast the fibroblasts are growing rather than other fibroblast lines, because they're all primary cells, and so there could be some differences there with passage. Probably the more interesting question is, why is this such a problem in iPS cells? We can have human beings running around with ring chromosomes, and I suspect it's just because one has so many divisions that are occurring in these cells in culture that you can't sustain a ring chromosome for very, very many passages, and so they're lost in culture in a way that they're not lost in humans. But that's another question that will need to be addressed. Hopefully we could do this in models as well. Yes?
So is there phenotypic variability among individuals who are affected by ring chromosomes? Because if there is a loss of heterozygosity, we would expect that you should get all sorts of things expressing.
There's a lot of phenotypic variability in patients with ring chromosomes. For one, it can be secondary to the loss of genetic material when the ring forms. Others, it's because there is mosaicism among patients that have ring chromosomes, so some normal cells, some ring chromosomes, some cells in which there are breakage products. A lot of it just depends upon the chromosome that's involved. And as I described, ring 14, it's a very interesting story, because whether there's deletions or not, the phenotype seems to be the same. There seems to be something, because of the ring 14 formation, that may be silencing other genes on that ring chromosome and may be contributing to the phenotype. So it depends upon the chromosome itself. Perhaps it depends upon what genes are brought together during the ring formation. There could be position effects, and it probably certainly depends upon the chromosome itself, whether there's haploinsufficient genes on the ends, or whether there are genes like ring 14 where there's suppression or some other mechanism that's causing that to happen. Yeah, Phil?
Do you have any idea how frequently disseparants are happening?
At the moment, we don't. And at the moment, we don't. We haven't tested that. Again, I think we're going to need to purify in some way what's happening. We can look at what happens after the ring forms by purifying those cells and then having those cells to examine later at some point. I think that's what we're going to have to do. Okay. Thank you.